Welcome to Advanced Anatomy Physiology. This unit we're going to be covering genetics, and we are going to start with some basic information here. Genetics is the study of our genes, and we'll get to what those are in just a minute. And there is a difference between the terminology genetics and hereditary. Hereditary, as you can see here, is the passing of that genetic information or the genes on to your offspring, whereas the genes themselves, as you can see, is the basic functional unit of hereditary. Now, we got to go back to Unit 1 and remind ourselves that what do genes do? They're going to code for specific proteins. And so everything we're really talking about with genetics is the study of genes or the coding of proteins, which makes us all unique, and all these proteins do different things for our body. And then also, as we talked about in our last unit with meiosis and gametogenesis, the hereditary and passing that information along. So if we look at our chromosome, if you will, you can see here this you have homologous chromosomes, which means that they are the same, one from mom, one from dad. And you can see that there's different areas on the chromosome which we would consider to be our genes. And typically, basically, the homologous chromosomes should have the same areas, if you will, of these genes. They may not be the same genes, if you will. Example, one might code for brown, one might code for blue eyes, and then one of them may be dominant, as an example. And that would be reflective of what eye color you would get. But this just kind of gives you an idea of what we're looking at here. These little bars and these little different areas here are the genes. And not every part of a chromosome is going to be considered a functional gene, if you will. Part of the chromosome don't code for any proteins at all. So I don't want you to think the whole entire chromosome is going to be coding for a particular protein necessarily, but certainly, as you can see with this picture here, you can see that the genes are matching up, if you will, one from mom, one from dad. And again, you can see, just as an example, dad might have code for brown, while mom might code for blue, and we have to just determine which one of those is going to be more dominant, and then obviously portray that, if you will. So when we look at chromosomes, as we talked about in previous units, chromosomes are found in both sex cells and somatic cells. So remember, a somatic cell is a body cell. It has 46 chromosomes, 23 pairs. You got one set from mom, one set from dad. And these are considered to be homologous, meaning that they are the same chromosomes, if you will, but they may not code for the same exact genes. So out of these chromosomes, out of the 23 pair, 22 of them will be considered to be autosomes, which express other traits, where the 23rd pair is the sex chromosomes, and that could be two X's, it could be an X and a Y. So just as a kind of a reminder, and if you had two X's for sex chromosomes, you would be a female. If you had a X and a Y, you would be a male. And so that's really determined by what sperm is actually fertilizing the egg, because the egg is always an X chromosome and the sperm are going to be either an X or a Y. So the male sperm really kind of dictate whether it's going to be a male or a female, depending on their chromosome that they're giving up during that fertilization. And so this whole process, this whole thing that we're looking at, is the human karyotype, which is the diploid genome, if you will, one from mom, one from dad, which creates this 46 chromosomes, our genetic or our DNA makeup, if you will. And you can see here in this picture all the different chromosomes here that we're slowly evolving and finding different genes and what they do. And if we look at these genes, we have a lot of terms that mean a lot, and they can get a little confusing for some people. So the alleles, or alleles, if you will, alleles I usually call them, are the matched genes on the same locus or location on the homologous chromosomes. So as an example, like we just went over, coding for a specific trait. So if dad's, go back to that slide here, if dad has brown, mom has blue, this is considered to be the allele, okay? And you can see that these are the matched genes on that homologous chromosome, and they should be in that same location or the locus, if you will. Now, if these two traits, alleles that are coding, are the same, if they were brown, if you will, they're homozygous. And if the two traits or alleles are coding differently, like we saw with the example there, they're heterozygous. So there's some differences between those alleles, depending on what we see there. So when we look at the genotype and the genetic makeup, we have either dominant or recessive, and typically dominant is represented by a capital letter here, and recessive is represented by a lowercase letter. And so what we have here is two capital letters. This would be dominant. If we have a little bit of both, typically the capital letter is dominant over that recessive. And if we have both of them lowercase, that would be 100% recessive. So this is the genotype, the genetic makeup, but now how is it expressed? And when it's expressed, that's what we call phenotype. So we might say, as an example, the genetic makeup or the genotype is for brown eyes is dominant. It's both of these as an example. And so then how that would be expressed would be that it would be brown eyes, and that would be considered to be the phenotype. Okay. So again, how is it being expressed? There's two different things that you need to look at, because when we get into this, more so with lab, because we have a lot of lab examples and case studies and things like that, but you know, you'll get the information that you need and determine what is this genotype. Is it dominant? Is it recessive? And there can be even a mixed bag of things, and we'll get into that in a minute. And then how is it going to be expressed? What would you expect that to be expressed as? Okay, so a genetic carrier is where it's an inherited the trait, if you will, but does not display the trait or show the symptoms. So you may inherit that trait, you may be a carrier of it, but it's not actually phenotyping or showing or expressing that particular trait, if you will, and that's certainly the case. So that can be passed down. You could be a carrier of it, but it may not be expressed. So you may have a little, you know, say you're completely brown-eyed and brown hair, but you still have a little bit of that inherited blue-eyed trait in there. You're a carrier, but it may not be showing, if you will. So as we were talking about with these alleles, dominant one allele masks the other allele. Okay, so obviously you would expect that. So as I said, the dominant is usually expressed with or expressed as a capital letter, and recessive, the allele is masked, and that is usually represented by a lowercase letter. So if you look here, if we have two capital letters, because they're the same, that would be homozygous, and it is dominant because they are both capital letters. Now if you have a mixed bag, because they're not the same, that's heterozygous, and it is dominant because of the capital letter. And if you get two lowercase, they're the same, so that's homozygous, and because they're lowercase, they are recessive. So we're really taking this dominant, recessive, homozygous, heterozygous, and combining them together to get this right here. So this is typically how you want to be reviewing it, is this last little part. So we kind of went through all this stuff up here with alleles, homozygous, heterozygous, and then also with the genotype, whether it's dominant or recessive, and just kind of combining that together. And remember, going back to meiosis 1, when we had our crossover, we had the during sex cell formation, we had the DNA that was crossing over, as you can see right here, and we were swapping genetic information. And then we had the variability, where they were what we call independent assortment, where they were just kind of randomly aligning, and this created variation as well. It was very randomized. So just kind of recapping that, we ended up creating 4 unique cells that were genetically different because of all this that happened again in meiosis 1. So again, the first bullet there is exactly what I went over. The gametes, the sperm and the egg, the genotype and the phenotype are very variable because of the crossover and the independent assortment. And so think about that, and then you get this random sperm and this random egg that are going to fertilize, and we don't even know, which one it is going to be. Males ejaculate like 7 million sperm, and so, it just takes one of those to fertilize, and so you can imagine the variability and the chances that we would get, what we would get, and that forms a zygote, your very first cell, which has 23 chromosomes from dad from the sperm and 23 from the mother. And so everybody is different, obviously, except for identical twins who share the same DNA. So the combinations are endless. That's the reason I have this mega millions here, because what are the chances, right? There's just so many different combinations that are involved. If we go back to Unit 1 and talk about all the different nucleotides that make up DNA and the combination of them, and knowing that we had all this variability, and we have hundreds of thousands to millions of nucleotides that make up a DNA and what order they're in, and how we get some from mom, some from dad. I mean, it's the numbers are astronomical. It's kind of like playing the lottery, right? The chances of winning, the odds of that. So, but we're all winning with this. Don't worry. Okay, so we'll get into some of this stuff here now. Dominant recessive inheritance. So the interaction between the dominant and recessive alleles. So we have monogenic trait, which is characteristic of just a single gene pair. And so what we want to do is take the genes, if you will, and put them into what we call a Punnett square. And this kind of gives us a possible gene combination and a possible outcome, if you will, of what we might expect for this offspring, or what to expect with that. So you get a lot of this in lab, obviously, but this is your basic dominant recessive Punnett square. And so this would be one example up here where you have the dominant recessive. Down here is the dominant recessive for that as well, and from mom and dad. And so if we take this capital letter B, this is our first square. If we go over with this capital letter B and down with this lowercase b, you can see that. And then same here, this big capital B down here, lowercase b, and then finally the 2 lowercase b's down here. So when you look at this, we have 3 dominant situations here and only 1 recessive. So the chances, as you can see here, the brown eyes are at 75% chance that this offspring will have brown eyes, whereas there's only a 25% chance that the offspring will have blue eyes. So I mean, this is a great example, though. I'm brown-eyed, brown-haired. My wife is brown hazel eyes and sandy blonde hair. All of our kids have brown eyes except for one child, and he has blue eyes and sandy brown hair. Now my mother has blue eyes, and my wife's dad has blue eyes as well. So we did have that in our genes, if you will, and so everything aligned just perfect for that to happen. But the odds were against us that we were going to have a blue-eyed child, but there still is that chance that could happen, and that certainly did happen here in this case. Okay, so you'll get more work. You're not going to really get you're not going to get work on the test, I don't believe, with a Punnett square in lecture. Don't quote me on that, but it's more so for the lab, a lot more hands-on with the lab. Okay, so now we look at some differences. Everything that we talked about here was pretty basic, dominant, recessive, but now we get into some things that are a little different. We have, our first one is called incomplete dominance. So normally we would have either the recessive being dominant or the recessive masking the dominant or the dominant masking the recessive, and here we get incomplete dominance, which is where we get heterozygote has an intermediate phenotype. So remember, phenotype is the way that it's expressed. Okay, so it's an in-between, if you will, between this homozygous dominant and this homozygous recessive. So here's an example. Here's the homozygous recessive right here is a sickle cell anemia. So if a person has homozygous recessive, 2 lowercase s's, they have sickle cell anemia. That could put them into what we call a sickle cell crisis at any time. So sickle cell anemia is where there's a defective hemoglobin on the red blood cell, and this causes issues with transportation of oxygen and could cause death if they have a crisis, which is basically where their red blood cells start to rupture and they can't transport oxygen. They have heart attacks and strokes and things like that. Now, there is an example here of sickle cell trait. So this is what I wanted to get at here, is where they have an incomplete dominance. So in this example, they end up phenotyping, if you will. This is the intermediate phenotype. They make healthy hemoglobin along with sickle cell hemoglobin, so it's in between. So they have this trait, and they're not healthy. I mean, they make sickle cell, but they also make healthy ones. While back here, this homozygous recessive, they only made sickle cell anemia. So there's a difference there. This is kind of this intermediate phenotype, this incomplete dominance. Not one or the other is dominant over the other, which normally we would see that, right? Normally if we had this capital S right here, that would be dominant, right? And that is not the case here, because we are clearly making healthy and unhealthy hemoglobin, so that's considered incomplete dominance. Now, multiple allele inheritance is another one where more than 2 allele forms, and what we get, we each receive 2 of these, but this is seen with ABO blood typing. Okay, and so what we see here is we see the capital A, capital I, and A, capital I, and B is the codominant. Okay. And the lowercase I is recessive. And so in this case, you are either going to be A, B, so these are 2 A's right here, dominant, homozygous dominant, heterozygous dominant right here, okay? And same thing, this is B, homozygous dominant, heterozygous dominant, and then we get this I here, which is going to be recessive, and then we get this codominant. So it's a combination of A and B together, which is considered to be codominant, and this is where you get your AB blood. So we have either A, B, O, which is O here, or AB, which is codominant. So you get a little bit of both, right? And that's what we see with this multiple allele inheritance. We get this codominant situation that's happening. They just can't determine which ones would like to be more dominant, so let's just add both of them to that mix. Okay. Sex-linked inheritance, so the traits are determined by genes on the sex chromosomes. So like I was talking about earlier, males, the sex chromosomes are not homologous. Okay, so you can see that if they were, I should say with the female it is, but not with the male. So with the female they have XX, which is homologous, they're the same, but male have X and Y. And so these chromosomes, therefore, on the male are not homologous, they're not the same, and they are different. And on the X-linked chromosome for both male and female, there are about 2,500 genes that trait for some things that you're familiar with: testosterone, clotting proteins, cone pigments, which would be in your eyes to give you color. And then on the Y, there's only 78 genes. But what's important about the Y is it has a sex-determining region on it, which switches on for the maleness. And this is why, males are males essentially, is because of this Y chromosome. It's what's switching on these male traits, if you will, where the females don't see that. So we know that, like we just went over, the traits are on there. If the male, if the X is defective on the male gene, the male will have a disorder, if you will, like hemophilia or color blindness. But the female needs to have two recessive XXs, if you will, to have a disorder. Okay, so it's basically the odds are against the male because they need to only have one X to have this problem, where the females need to have both being recessive. So these are passed along, these sex-linked defective genes, if you will, are passed along to son by the mother, as she is the X chromosome carrier. So remember, it's on the X chromosome, so males are XY, so the Y came from the dad, can't come from the egg is X, and so because the egg is X and it came from the mother, that's why it's passed along to the son by the mother, as she is the X chrome carrier, if you will, and the father can only donate the Y. So if a male has hemophilia or color blindness, it's passed along from the mother to the son. Now the mother can pass along these problems, if you will, to the daughter, but both of the X's from the mother and the father must be recessive. So that's kind of what we're getting right here. They need to, female needs to have two recessives in order to have these disorders. So that is a little bit from mom and a little bit from dad in that situation, and they both must be recessive. Okay, last couple things here. We have polygene inheritance, or the polygenetic trait. There are several gene pairs in different locations working in tandem, so they're not on the same locus, if you will, right? The same location. These are in different locations or locus on the chromosomes, and they eventually result in a different phenotypic variation, or how they are expressed differently. And so this is polygenetic trait. It's the characteristics of multiple gene pairs. So an example would be skin color, height, metabolic rate, intelligence. These are all coming from different areas of our genes that kind of are put together to create this very specific phenotype variation, if you will. So look down here just as an example here. Your skin might, this is just one example, your skin might take all three of these different gene locations right here and create either this ABC, capital letters, or ABC, and that's going to be a difference in your skin color as an example. And again, they're pulling from different gene or gene locus areas, if you will, to express this. And lastly, just some chromosome disorders here. Aneuploidy is the missing or too many chromosomes in a part of the set. So some examples would be monosomy, which is only one chromosome. And an example of monosomy would be Turner syndrome, as you can see here. So they only have that X chromosome. Trisomy, they have 3 chromosomes. They have an extra chromosome on the 21st chromosome, and that would be an example. This would be Down syndrome. So you can see usually people with Turner or Down syndrome have a specific unique look to them, and you can see that in these photos right here. Polyploidy, extra sets of chromosomes. Now, when you have too many chromosomes within a whole set, now the humans will not survive this. Typically, there's programmed death that will occur if the chromosomes in the cell have too many chromosomes. But this polyploidy can happen in plants, as they have their DNA as well. But typically in the humans, this won't go too far, because there's a natural programmed cell death if this were to occur on that as well. So this is genetics. If you have any questions, let me know. We're really kind of going over the basic information here. And more just kind of terminology and what that means. And the Punnett square stuff is more geared for the lab, and there's a lot of examples in the lab and case studies that you fill out with your worksheets. But for now, just kind of understanding what is dominant, recessive, homologous, homozygous, heterozygous, what are the differences between incomplete dominance versus multiple allele inheritance, sex-linked traits, dominant recessive inheritances, and finally polygenetic traits or polygene inheritance. So if you have any questions, make sure you make a list. That's what I always like to tell people, to make a list of all these things and write down what are the differences between each, so you can see them on a piece of paper. I think that will help out quite a bit too, because you're going to have to know those obviously for the test, and what does one mean versus the other, and I think getting a better visualization than just looking at the PowerPoints will help you with that, and also in your own words. So if you have any questions, let me know. Other than that, this concludes genetics.